So the rationale behind our study was the following. We actually wondered why certain patients receiving CAR T-cell therapy respond very well to the therapy, while more than 50% of the patients do not have a very good response and actually relapse very soon. And we were wondering whether we could define biomarkers already at the time of cell preparation, at the time of apheresis, which would actually predict the response to therapy. So the study itself was a study including 33 patients. 7 patients dropped out of the study, so we could analyze actually 26 patients. So it's not a too large study. However, it was prospectively performed, and we monitored the disease outcome over 12 months, and it was the endpoint of the study. So the most important finding of the study was actually that we could identify at the time point of apheresis, so before actually going into the very laborious process of manufacturing the CAR T cells, which is not only laborious but also very expensive, identified biomarkers on patients' T cells, T lymphocytes, which will tell us with a certain certainty whether these patients will have a benefit from that very therapy, or whether the benefit will be rather very low, and that actually before waiting for the CAR T-cell therapy should be better treated with further treatments if that would be possible at that stage of the disease. Yeah. So one implication of our study is, so we identified actually the marker combination which we identified is actually CD27 and CD28. So these are two very important co-stimulatory markers on T lymphocytes. And in the case that these markers lack on the majority of T lymphocytes, the future outcome of CAR T-cell therapy will be very poor. So what one can do in order also to serve and help the patients who have a very high ratio of these CD27 negative and CD28 negative T cells would be, for instance, to enrich for the remaining CD27, CD28 positive T cells in the apheresis product of those patients before going into the manufacturing process for CAR T cells. And that could maybe be a game changer, because then the starting population for manufacturing the CAR T cells would again be like the one of patients who can actually respond very well to the therapy. So in other words, by a possible sorting process, one could actually kind of rejuvenate what is in the manufacturing process, and that could be a big game changer then for patients. The other side of the coin would be that we would have a rational decision point actually to say who should get the therapy and who will certainly not benefit from the therapy if it is done in the way it is done right now, transducing bulk T cells. And if these bulk T cell preparations contain a large number of these CD27 negative, CD28 negative T cells, then the outcome will be poor, and the recommendation would be not to do it in that way for these kind of patients.